Welcome to this breakout session. It's great to be here in Vegas. Every 3 hours in Vegas for a big sporting event like Super Bowl, 200 million bets are placed. In those same 3 hours, 130 million tweets are sent. That's a lot of data. I want to share one more data with you. 2,124. That's the number of people who die of cancer in those same 3 hours. We at Thermo Fisher Scientific have made a pledge to make this disease a chronic disease, so it can be cured just like common cold, and nobody will die because of this horrendous disease. I'm Puneet Suri. I work for the Life Sciences Group in Thermo Fisher Scientific. I'm based out of Bay Area, California. There are only 3 things you want to know about me. I look much better in person than the picture on the screen. I dream big, and I make it happen. And my goal is to use technology to advance science. I head the software for the genetic analysis division and also spearheaded our cloud initiative. Why do we all come to work every day? We all want to make a difference in this world. This is Jamie, a mother of 2. A few years ago, she got lung cancer. Just like any other lung cancer patients, she underwent a surgery to remove her tumor. Like most of the lung cancer patients, after the surgery, they're sent back home. Very high percentage of them come back within a year or 2 with a stage 3 or stage 4 cancer. And at that point, it's a death sentence. Jamie was lucky. Her doctors knew about this Provenio lung RS test from our company. This test can tell you if the patient is going to be in a high, medium, or low risk category for reoccurrence of this cancer. And just after her surgery, they did a test on her and found out that she was in a high risk category. And instead of sending her back home, they put her through chemotherapy. And I want to report to you today that she is cancer-free today. Here are a few other examples of our company making an impact. James Bain was in prison for 35 years in Florida. And in 2010, he was released from prison after DNA testing proved that he never committed the crime he was convicted of. I wish this technology was there 35 years ago. Another topical topic these days is Ebola. More than 5,000 people have died in the last 9 months due to this disease. Just a few months ago, FDA approved one of our instrument platforms to be used for testing Ebola throughout the world. Thermo Fisher has been in news a lot. We are the technology and science behind the headlines. Another example of making an impact is H1N1. H1N1 was a big epidemic in this country in 2009. More than 61 million cases were reported to CDC. More than 12,000 people died in the United States alone. And we were at the forefront in fighting this disease. Again, FDA approved one of our instrument platform, which was used throughout the world to test H1N1. This is how we strive to improve human condition. Do you know every year 6 million people die of cancer? And we wanted to reach not to just one Jamie, but every millions of Jamies across the world. How do you make this happen at a larger scale so you can touch millions of lives? If I do a DNA sequence of any one of you in this room, it'll produce 3.2 billion base pairs. What does it mean? It's ATCG in a sequence. And if I put that on a ticker tape, it'll go from San Francisco to New York. But to get any meaningful results, I probably have to sequence you 20 times. And that ticker tape will go around the universe or the Earth 7 to 8 times. That's the amount of data we're talking about. Just imagine the magnitude of data. Now, to do any meaningful discovery, we need to do real-time analysis of huge data sets to decode genomic information. We need ability to store and manage these large scientific data sets. We want highly interactive visualizations for our scientists so they can manipulate this data in seconds. They're like, sometimes these scientists are looking for a needle in a haystack, and we want to provide tools to them so that they can do this discovery really fast. And also, they want to share and collaborate with other researchers across the globe. And what were we doing a few years ago? We were shipping desktop-based solutions. But you all know the hassles of OS upgrades, application versions, etc. That's a big hassle we don't want to deal with. These desktops have limited compute and storage capabilities to handle these complex and large data sets. There were no easy way to share and collaborate with each other. And there was no effective archiving and backup and security. If you're carrying a laptop on your shoulder, there's not much security at that point. And we wanted every scientist, a better way for every scientist. We wanted to give them unlimited storage, compute, memory, and scalability. Essentially, providing every scientist and researcher across the world a supercomputer on their desk. That's how our vision was born, to create one of the best life sciences cloud platform. We wanted to create an ecosystem where all our instruments, after they finish the run, push the data to the cloud. Not only push the data to the cloud, but also can talk to each other, just like Internet of Things. We want a place for scientists to collaborate, share this data effectively and securely. We wanted to create a SaaS platform, an applications store for scientific applications, where applications not only from us, but from you guys, and also our third-party partners can put those applications there. We want to link to our e-commerce sites so that you can subscribe and buy these apps, not only these applications, but scientists can also buy reagents and other consumables to do their experiments and studies. And I also wanted to connect to the external data sets like NCBI, CDC, and so on, so that we can accelerate discovery and research. Earlier I told you I dream big and I make it happen. Let me walk you through the make it happen part of the story. What does a scientist does in a day? He gets a patient sample. He runs that patient sample on our instruments, gets some results, analyze them, view those results, and trying to find some meaningful insights. What is he looking for? He's looking for what's causing cancer. What drugs will work? Is therapy working? This sounds simple, but not really. There are huge challenges to make this a reality. Our existing solutions can't even address these complexities. Customers use Excel, which is cumbersome and not optimal. Sometimes they do multiple smaller studies, put their data in Excel, try to manipulate through that. Sometimes they use multiple tools to get insights. Sometimes it takes days instead of hours. And some of the complicated analysis workflows are not even possible with our current solutions. So if you talk in terms of computer science, it boils down to 4 dimensions of complexities: storage, compute, performance, and scalability. For storage, we want ability to store millions of records. We wanted to have a real-time analysis of these huge data sets. That's compute. For complex visualizations, we want response time of few seconds instead of hours. And we want a cloud platform which will scale to thousands of users and millions of studies. Let me walk you through the journey we took in resolving these complexities. The first step of our iteration in this journey was to start with a reference architecture. This is a typical reference architecture for a transactional web application. You can see we have load balancers in front of web servers, app servers, And we started with the relational database as our persistence layer. And we knew that this is not going to work for us. It's not going to scale to the number of users and increase in data and give us the query performance we're looking for. We don't want to deal with sharding and resharding. That's a big hassle. We wanted a flexible schema so it's easy to enable additional workflows in the future. Today, the science is moving really fast, and scientists will be using cross-analysis workflows in different domains. So we wanted our schema so that it can handle these additional workflows which you don't even know today in the future. So that took us to the next step of our iteration, which was to identify scalable storage solutions. And NoSQL was the way to go. We picked DynamoDB. Most of you know the advantages of that in this room. It's a managed service from Amazon, so there's no administration overhead. The query performance was great, even if you add billions of rows. It supports flexible schema for us to support futuristic domains. And it has extremely fast read and write in order of a few milliseconds. And this is how our architecture was with DynamoDB. So we took out the relational database and put in the DynamoDB. Some of those things worked really well with DynamoDB. It's a managed service with flexible schema. It can scale even if you add billions of rows, and it has extremely fast read and write performance in order of milliseconds. And I already said it's a managed service, so there was no additional administration overhead. Let me pause here and talk about a way how we store this information in a project, which is a study. This is a high-level logical model of our data, and we talk in terms of project. So what's a project? Let's say we're doing a cancer study across different population. That involves thousands of samples, patient samples, thousands of genes which we are screening for. And the project contains all this information. It contains instrument runs, which we have to run for those patients. It involves patient samples, the genes we are screening for, all the raw signals coming out of those instruments, and finally the analyzed results. So when we went to Dynamo, it partially met our storage and query needs, but there were some gaps. The gaps were, we faced a hot hash key issue. Since we were saving thousands of related records like raw signal with a common hash key like instrument run. So performance was not optimal. We were getting like tens of seconds instead of few seconds. There are close to 1 million records which has to be read or written at the same time. And with the batch size limitation, even with parallelization, this was a bottleneck. For attributes for an instrument run, some of these attributes were bigger than 400 KB. So we have to split and save them in S3. When we started this project, the limit was 64 KB, but now it's 400. But in spite of that, we have to do the splitting and move information to S3. And for larger studies, we need really high read and write capacity in order of thousands, which resulted in, it was not very cost optimal at that point. So that took us, so what do we really needed? We needed a solution which can store large numbers of related objects. It's cost effective for read and write operations for large data sets. It has no batch size limitation and have an ability to query into large data sets very efficiently. That took us to the next step of our iteration, was to identify scalable storage solutions for large data items. And we evolved our architecture to add another layer of storage. Now we have Dynamo and S3 as our persistence layer. If you look from a project perspective, the stuff which is smaller objects, like instrument run, patient samples, and genes, were stored in DynamoDB, and larger related objects like raw signals and analysis results, which are in millions, were stored in S3. And this met our storage needs for performance and cost, but we have to do a few more things for query. for both performance and cost. So I just said that in the last slide. This slide is just reiterating the same thing. DynamoDB was used to smaller unrelated objects, which will grow to a large number. And S3 was used to store larger objects like raw signals and analysis results. And all these objects were saved as Amazon S3 object using a Google protobuf. And we also felt that for these large data sets, S3 was quite cost effective. But there was another thing. We wanted to have a really fast real-time query of these complex visualizations, which are highly interactive. Some of these things which you're seeing on the screen have millions of data points, and scientists are manipulating this thing in real time and expect the results to be back in few seconds. So we are going to the cache, fetching the information, and replotting these things in few seconds. This is very different than the batch processing which happens in typical web applications. So what did we need? We need complex visualizations which requires gigabytes of data to be queried in few seconds and presented to the user. These visualizations, as I said, were highly interactive, which requires constant update of data. So we need extremely fast read and write times. And we want to support, of course, we wanted to support concurrent users without any compromise to our query performance. That took us to the next step of our iteration, was to identify solutions for fast real-time response and queries. And we know the in-memory storage was the way to go. We need to have really fast read and write to enable this fast response time. And read and write for S3 was not optimal at that point. So we used elastic cache as in-memory storage on top of S3 and Dynamo. All related objects in S3, which are frequently accessed by the customer, were maintained in elastic cache as an individual records. And indexes were created in Dynamo so that these can be retrieved easily from elastic cache. And if you look at our architecture, now you have 3 layers of persistence: Dynamo, S3, and elastic cache. And if you look from a project perspective, you have, again, the smaller objects were saved in instrument, like instrument runs, patient, and genes were stored in DynamoDB. The large related objects like raw signals and analysis results are saved in S3, and the objects which are accessed by customers very frequently, those are kept as records in elastic cache. And the indexes for them were kept in DynamoDB, so based on query patterns, we can retrieve them really fast. And at this point, we met our storage and query needs for performance and cost. But that was not the end of the story. We wanted an ability to analyze huge, larger projects and studies containing thousands of patient samples, and we wanted those results to come in few minutes instead of days or weeks. And we wanted a solution which is scalable to support thousands of concurrent users doing big studies. And we wanted our existing desktop algorithms to be optimized for parallel analysis of this data. Let me step back and talk about what do we ship on a desktop. As soon as you reach about 200,000 data points, desktop will run out of memory. It kind of crashes. So it's not even possible for our researchers and scientists to do big studies. They would create multiple smaller projects on studies, analyze them, bring them together in Excel, and manipulate the data in Excel, as you can see in the next slide. I know a researcher who has about 10 to 15 feet long Excel, in which we have got huge number of studies, put it there in Excel, and try to manipulate the information there. We don't want our researchers and scientists to spend time in Excel and go through that nightmare. It's our obligation to give them answers in few minutes instead of hours or days. That took us to the next part of our iteration, which was to identify solutions for real-time analysis of this data, huge data sets. And we knew EMR was the way to go. It was used to perform real-time analysis of huge data sets. Any small analysis was done in-memory. And we updated our existing algorithms to derive massive parallelism using Hadoop MapReduce framework. As the data was already in S3, S3 was used for input-output instead of using HDFS. And since we knew what were the bigger jobs and what were the smaller jobs, EMR was spun off and shut down based on the needs and was quite cost-effective. Today, if the scientists, which I just talked about a few slides ago, have to do that on a desktop, probably it'll cost him weeks or a month and thousands of dollars by the time he gets an answer. This, you can do it in $1. That's the power of cloud. Now our architecture evolved. Now you have 3 layers of persistence, Dynamo, S3, and Elastic Cache, but you also have EMR to handle these big analysis jobs. Now, let me summarize it, how our project looked like. As I initially said, the smaller objects like instrument run, patient samples, and genes were stored in DynamoDB. The large related objects like raw signals and analysis results were stored in S3, and the ones which are accessed frequently by the customer have a records stored in Elastic Cache. Indexes are maintained in DynamoDB. Now, when the big job comes, we know what the big jobs were. We spun off EMR, and if it's a small job done, we goes through normal in-memory analysis. And after that, the results are written back to S3 and Elastic Cache, and they kept in sync. Now, we met our needs for performance and cost for both storage, query, and analysis. And I showed you this graph a few minutes ago. If you look at it, as I said, desktop will crash at about 200,000 data points. With cloud, now you can run millions of data points, means the researchers can do thousands and thousands of samples in one study, and they can go for a quick cup of coffee, grab a sandwich, and the results are there. That saved thousands of dollars for our researchers. More importantly, now they can spend their time doing scientific discovery, not worrying about Excel. And we got more than 10x improvement on this thing. That took us to the last step of our journey. That completed our journey. But this is just a beginning, and we'll improve as we move forward. Here are the few learnings. Every use case is different, so there's no one-stop solution. I have to say a special thanks to our AWS team for their excellent technical support through this whole journey. They were with us to the inception, to the deployment. We just launched this platform a couple of weeks ago, and without their help, we would have not been able to achieve this. So with a cause, an excellent team, and a great support, anything can be achieved in a really short period of time. This journey started about 15 months ago, and with the help of them and the whole team at Thermo Fisher, we've been able to fulfill this dream. I've been talking for the past 25 minutes. I want to show you a demo so that you can see it for yourself. For that, I'd like to invite Shakila on stage. She's our cloud application lead for the last 2 years and extremely instrumental in making this dream a reality. Please welcome Shakila.
Can I get the clicker? Okay, thanks Puneet. A little bit about myself. I manage the cloud applications group for Puneet. I love hiking. Any hikers in here? I really love hiking, but I love alcohol too. But, but I'm pretty sober today. Not tomorrow, I'm very sober today. And I'm also a co-founder of a non-profit group. I'm very proud of that, with the cause, child literacy. So that's a quick overview of who I am. You can come outside and talk to me about anything, including alcohol. How to get into your genes? Boy, that's hard, right? You guys know it's really hard. But I'm talking about human genes here. So let's... Okay, I'm just going to walk through the workflow of what a scientist would do to actually understand how your genes work and what's the process, right? So you take a patient sample and you take a normal sample. Right now we have 30,000 plus genes identified in the human body, which you know has some expression levels based on your diseased or not diseased. So the first step the scientist is going to do is look at those 30,000 genes and see if there are any significant subset of those genes which are actually different compared to a cancer patient and a normal person, right? That's the subset that they want to research more on and do studies more about. So once they find that subset and trim down like 20 plus genes or whatever that number is, they're going to do population studies, because the way the genes work in Hispanic population could be different from African, American. So you got to find a distribution and see what is that final subset of genes which is normalized across the different population. So once they have that set of information and understand how the genes work with each other, then they can actually do real meaningful things like, okay, what kind of therapy should I give this person? What kind of drugs will work for this patient? And what are the different types of cancer? You have lung cancer, but there are different types in it, right? All that analysis and research can happen once this information is there with the customers. So what I'm going to do is just talk about how they take these hundreds of genes and narrow it down into a small subset. I'm going to give a quick demo of the application. So that's where we've hosted our application a few weeks ago. It's live right now. So let me switch and show you the app. Okay, can you see that? Awesome. So the scientist has come here and created some projects. So they're going to open a project. I've called it Amazon Demo, but that's not usually the name. So once they have loaded all the information into the project, what is happening right now is the project is being loaded. It's reading the information from S3 and putting all that information in the cache, so it's available for this user, so they don't have to go back to S3 all the time to get the information. So it's reading your plate information, the gene information, and the sample information. So what does this project actually contain is very similar to what Puneet said, all the signals that came from the instrument. Now you got to put everything together and analyze to see if it makes sense to the scientist. So there are two steps I'm going to talk about. What are they going to do? They're going to analyze their data, put everything together, use the parallelized algorithms and analyze and get some results. And then they're going to walk through the results to find that significant subset of genes that they are interested in. So when they analyze the data, it's going to fetch everything from cache. Again, it's not going to read from S3, some elements from DynamoDB, everything is in cache. And based on the size of the analysis job, it's either going to do in-memory analysis or launch an EMR Hadoop cluster. In this case, it was in-memory analysis. And then what are they looking for, right? They're looking for visual patterns, as I mentioned before. So let me show you an example of that here. So what is this? Is a heat map of how the genes are different. So now you can clearly see that this segment of the samples are the patient samples. You know, they're called as the ones who have seminoma cancer, right? This is one of our customer data. And this segment of the samples are the genes that are not much expressed, but you can really clearly see that these set of genes are highly expressed, which means there is a difference, and this is a set of genes you want to take and do more research on. So this is what a scientist would do on a day-to-day basis. Today they have to do all this in Excel because they get the data, they export it and map it and do all the translation on Excel. But here in the application right now, they can do it in a few hours. I mean, I don't have a big project, but usually they can do it in less than an hour. There are a lot of other means that they can get these visualizations. So when I go to each one of these visualizations, it's fetching all the information from cache. It's not going to S3, it's not going to DynamoDB, and that's why we're able to give it the few seconds of response time that's required. The second aspect I wanted to talk about was editing the information, right? So again, it's pretty similar. You go here, you have a lot of drill down workflows. They look at the instrument signals and everything. And if they want to remove something because they think it's a problem with the instrument and it did not analyze properly, they can go and select that. And what am I actually doing here is editing the information. And you saw it was like really fast. Purely because we save it into the cache and we let the user proceed whatever they need to do, and we asynchronously call the saves and persistence back into S3. So you don't hold up your customers while they're doing the analysis. So that's a quick demo. If you want to learn more about this, please come talk to us. I'm going to switch back to the slide. So that's a summary of the demo. So the scientist was able to successfully find the significant genes in the seminoma cancer patient that they're going to work on, and that was the list of genes. Customer feedback. So we went live in November, but we had a few iterations of alpha and beta releases for a few months, and we've gotten really good feedback purely around, as you can see, the number of samples that they can run. They were able to run like few samples before, now they can run thousands of samples, and they're very happy about it, which means more innovation and more discovery. The speed of analysis with all the EMR and parallelization, that's amazing. And of course, new visualization, and trust me, all our scientists love Mac, so they can access it from Mac or PC or Windows Surface or iPad or anything, so they're pretty excited about it. So we did get good reviews, so I'm very happy about that. And now I'm going to transition to Puneet. He's going to talk about the endless possibilities and the future vision of Thermo Fisher.
Thank you for giving a great presentation. It was a great demo. So let me talk about what do we see in the future. Today when you go to a doctor, what do? It takes your blood pressure, it takes your temperature, give you a cholesterol level, your blood sugar level, and based on those reports, if something is not right, they'll give you some medication and send you back home. And you go to the doctor next year again and the same thing happens. Do you really believe that's going to detect early stages of cancer? Absolutely not. So a few years from now, when you go to a doctor, you'll still get the same results, blood pressures, cholesterol levels and whatnot, but he will also get your blood sample and do a DNA sequencing in you. This is the same 3.2 billion base pairs I talked about here earlier. And now, since he has your genetic information, he can tell you what the possible health risks were and what's the drug response time on you, what drugs will work on you, what will not work on you. So that's going to happen, and this will be all saved in the cloud. So when next year when you go to a doctor, since all this information is in the cloud, he'll still get the 4 vital things I talked about initially, but he'll query the cloud and get your sequence. He'll probably get your blood sample again and sequence you again. And then if there's something is not right, there's a mutation in your genetic information. So something has gone bad. What is it? You got some preview of that in our demo that we can find and distill down. But now, since this information is all in the cloud, the doctor knows something is not right. So what he's going to do? He's going to query that information from the cloud, which is also connected to the public databases like NCBI, and diagnose the disease. Based on this information, he can now prescribe you the drugs which is going to work on you because he knows your genetic makeup. And if there is no drug available, he probably will give you a clinical trial which you can go, Because of technology, this is much more proactive and personalized medical care. And every one of us in this room will have access to it in few years' time, not the chosen few. And that day is not that far. This is what the personalized medicine is all about, and we are working tirelessly to achieve this goal. Thank you, and let's make this world a cancer-free world. Before I leave, I would also like to say thanks to all the teams in Thermo Fisher, the applications team, the infrastructure team, the platform team, the design team, the product management team, to help us achieve this goal and make our dream a reality. And without the help of our AWS partners, this would have not been possible. I'd like to invite Sami, who's our AWS partner in this journey, to take you forward.
So thank you, Puneet. Thank you, Shakila, both for the presentation, but also thank you for going through this journey. I think we all agree that the ultimate beneficiaries of this journey is essentially anybody, the entire planet. So thank you to the Thermo Fisher team. My name is Sami Zuhurdin. I'm a solution architect with Amazon Web Services, and I have essentially the privilege to be able to support Thermo Fisher as they go through this, as they've gone through this journey. What I'd like to do is sort of just turn this back around. I only really have one main point, which is you've heard this journey, you've heard this story, and for you to turn around and ask, what is the story that your data is trying to tell? All of you are sitting on some sort of data, some sort of data sets, or some sort of extraction from the things you work on, and how are you enabling your data to tell its story? I'd like to offer you some advice, some very practical advice, built on actually the lessons from Thermo Fisher, on what you can do to enable your data to go through a similar journey. Let's take the journey that Puneet talked about that a scientist goes through. They're essential, this is a fundamental process a scientist goes through, is to collect data, store data, analyze that data, and then bring some nice representative, some meaningful visualization to it so you can spot that needle in a haystack. And it's a common sense framework that makes sense for genetic research, it makes sense for drug research, it makes sense for any variety of types of research. In any space. But let's use it as a blueprint and take these discrete stages, collecting data, storing data, analyzing data, and visualizing data. Let's try to model your data problems in the same fashion. What I'd like to highlight is if you look at the big data practice on top of AWS, it actually breaks the problem down in the exact same way. Look at your data footprint and figure out how does your data go from the left to the right of this equation. Whereas before, maybe a few years ago, the idea of just having enough data was a challenge. Maybe data was inadequate, maybe there were sparse gaps in your data and you had to make assumptions. It seems like that problem has definitely shifted to the right. Now there's just an abundance of data. The number of papers that are being produced, the amount of sequencing data you have access to, whatever it is, data has exploded. It's no longer an issue of sourcing data, or it's no longer a constrained resource. What we need is a sophisticated way to ingest that data. So that first step in terms of AWS services, that first step for us is if you can leverage a product called Kinesis. Kinesis is a service for mass ingestion of data. That could be data coming off of instrumentation, it could be data coming off of collection devices, it could be data coming off of servers. But whatever that endpoint is, you need a reliable way to ingest hundreds of thousands, if not millions of points of data in a short period of time. That's what Kinesis gives you. It gives you a durable way to ingest that data and not leave any data on the floor, not drop any data on the floor. Now, as we continue forward, we need a long-term way to store that data. S3 is a great logical choice. It's sort of our grand central station of all our data sources. You can put things there. It's got 11 nines of durability. It never goes away. It's replicated several times. Now that that data is in S3, you've got a variety of processing frameworks on top of that data. Now we can try either running that data through a Hadoop framework in our managed Elastic MapReduce, which is our managed Hadoop framework, or maybe it's just that data is something that the insights are gained by large aggregations. Aggregations across billions of rows, billions of records, are intersections of that data with other data. That's where our Redshift data warehouse product comes in. And finally, we have our huge, rich ecosystem of visualization partners that allow you to take an offering from our marketplace and plug it into your analysis framework. Because ultimately what we want is some nice demonstrative way to look at our data, run some time series crunching, collide that data with other sets of data, and expand the scope and see if there's any insights we can gain. These are all partners that are available on our marketplace. Now the question becomes which of these? There's choices in every vertical. And the question becomes sort of which choice makes sense? That's not clear. If you look at the 15-month journey that Thermo Fisher had to go through, that was a lot of back and forth, a lot of iterations, a lot of what worked and what didn't work. You're going to go through the same thing. You're sort of going to have this recursive loop where you're experimenting to get data, but you're also going to have to experiment on your data to see what works. And this is the benefit of having an API-driven infrastructure. An infrastructure where you can say, I would like a data warehouse. I would like a bigger data warehouse. I would like 10 servers in a Hadoop cluster. You need to be able to have programmatic infrastructure to do that, because you can't simply sit there and buy hardware or iterate on your hardware if it's physical and you're always having to go through long, laborious processing cycles. That wouldn't benefit your data. You need an API-driven infrastructure to do that. And ultimately, you're going to come up with findings. Now, the thing is, what I think the journey that Thermo Fisher taught us is that they went through taught us is that it's actually not going to be one answer at the end. You're going to have a collection of answers that say, based on certain criteria, you're going to use infrastructure differently. And so instead of calling this programmable infrastructure, I call it programmatic infrastructure, where you're going to receive a job. You're going to take the data that's trying to tell a story within your infrastructure and understand, get to know it, get to know how big it is and how fast it comes in, where it should be stored, and then figure out how to match that up with the processing framework. And these answers are going to vary based on the job requirements, the job size, the velocity, the desired answer time. And you want to plug that into a framework. So what this does is actually creates a workflow. It creates a workflow around your infrastructure, which is what you're going to need to do. Big data problems are ones where you bring compute to your data as opposed to bringing data to your compute. And you have to innovate a lot. And you can't innovate unless you're willing to try different solutions. I think in the case of Thermo Fisher, it was great that they went through enough iterations to realize, you know what, if we have millions of, potentially billions of data points, maybe time series data, maybe sequencing data, those are very hot, that should be stored in a cache. It's a very practical learning. You know what, these are large sort of blob type objects. These are larger objects. These are read once and then reused many times within a single host. That's a great candidate for S3. This is semi-structured data. This has some semblance of structure to it. This has some semantics that we can put on top of it. It's a great choice for Dynamo. And so ultimately, you can use this as a workflow to overlay on top of your data. And so, well, based on the type of request, we'll choose the type of processing framework that goes through. So I'll end with that. I'll say thank you for coming, and then I'll invite you to sort of go back, use the lessons learned from Thermo Fisher, and then really use the ability to use a programmatic infrastructure, a programmatic API-driven infrastructure, to iterate on your data many times, and hopefully it's going to tell you a similar story to the one that Thermo Fisher ended with. So again, thank you for coming, and hopefully this gives you some ideas.